Thanks for the introduction, and good afternoon, everyone, and thanks a lot for the opportunity to speak here. Owen Edwards wasn't able to come out here. I think he might be remotely watching this talk, but he wisely decided to stick back in Western Australia instead of coming out here. So I'm going to be talking about genetic biocontrol of Cots, and first let me mention a little bit about my background. I have a lab at Macquarie University where we're working on a number of genetic biocontrol strategies, mostly in insects. So I'm definitely not a Cots expert. This has been a fascinating meeting the last couple of days, and it's where I've learned about 90% about what I know about Cots was at this meeting. So the point of this presentation really is for me to introduce some of the genetic biocontrol concepts that have been effective in insects, and then start having maybe more of a conversation about how this could apply to Cots, and whether or not it actually would be appropriate or not. So first, essentially all of the different genetic biocontrol tools that are out there, using synthetic biology, Wolbachia infection, or what have you, are inspired by the sterile insect technique, which was developed in the 1950s. And the way that the sterile insect technique works is you use radiation, this could be gamma radiation or X-rays more commonly these days, to sterilize some insects. And then preferably what you'll do is you'll just release males into the field. The males will go compete with the wild type, the sterilized males will compete with the wild type males, find females, mate with them, and then the females that mate with the sterile males won't have any offspring, and then that population will go down. Now, typically you want to just release males, and there's a couple of reasons for that, and with insects, and especially with insects. One is if you're releasing both males and females, both the males and the females will have been sterilized by the radiation, but instead of looking for the wild type females, the males will hang around with the co-released females and end up mating with them. And then the other reason is, in a lot of insect species, it's really the female that's responsible for most of the harm. So mosquitoes are the best example of that, where male mosquitoes don't bite, so you can release as many of those as you want without really without worrying about it. The first insect that this was applied to is the New World Screwworm. So the New World Screwworm is a pest in the New World, which was a major problem for livestock, especially cattle, and also to some extent sheep, I believe. And it would lay eggs into small cuts into these that these animals have, and then the larva would eat the flesh of those animals and cause just awful infections. I've spared you the photographs that most people show during these presentations. Pretty awful. And using the release of sterilized screwworm, in this case it was both males and females, they were able to successively push the screwworm out of North America, then successively out through all of Central America, and now there's this permanent barrier down by southern Panama where there are factories that are just churning out millions upon millions of these every week. They're distributed by airplane, and every now and then there'll be a little outbreak someplace north of the border, and then that'll be suppressed. So my point here is that these types of technologies have been used on really large scales, not for the purposes of completely wiping out a species, but certainly to extirpate it from a particular region. SIT is also something that has then been applied to a number of different insects. This includes medfly, Q-fly, colony moth, pink ballworm, tsetse fly. It's something that's used in Australia to control the Q-fly and the medfly. And the scales of some of these operations are really remarkable. So some of these factories that mass rear these insects will be making, say 20 tons of insects every week, and releasing those in, say, ports of entry, or to help suppress populations if there's an outbreak. Now, how might technologies like this be applied to Cots? So unlike these insects, you obviously can't be releasing lots and lots of males out into the wild, since the males are just as capable as the females of causing damage to the corals. So it's likely that the type of approach that would be necessary is you would use culling, as you do now, to reduce the populations as much as you can in a particular area, and then you would say stock sterile males. In this case, they've got engineered males, but they've been engineered to be sterile was the point. And then those males would interfere with the reproduction of any remaining Cots there. And of course, those released males would also have to be at a density that's below that density that's likely to cause harm. And there might be possibilities to either have them, released and sort of roam free, or alternatively, it might be more appropriate to have them caged, depending on how the different aggregations might work. Now, there's other ways to potentially combine this technology with other types of technologies that are out there. So in this example here, I'm imagining applying the lessons learned from the Judas goat approach, which was utilized to get rid of, I don't know, Mike Pence moment here with this fly on my head. So there's the Judas goat approach that was utilized to get rid of the invasive goats on the Galapagos Islands. And in that case, what they did is they had a radio tag on these goats, and since goats are social animals, the radio tag goats would go hang out with all the other goats, then a helicopter would come by and get rid of all of its friends. A few weeks later, that goat would find another group of goats, and then they would repeat that process. So kind of an unfortunate life for that goat. But we could imagine something potentially similar here, where you stock with these males which are not capable of reproducing. They're capable of suppressing the population, but you've also tagged them, right, with a GPS tag or something else, and then you can use them to help to locate mating aggregates during mating season, so you can find out where the other Cots are that might otherwise be difficult to find using some of the current techniques. So now, assuming that those ideas seem somewhat appealing, the question is, how would we actually implement a system like this in Cots? And in insects right now, there's a few different approaches that are used, and overall they're not really suit for purpose in Cots for a few different reasons. With the exception of, perhaps you could use radiation. I don't know if anyone's tried using radiation for sterilizing Cots. Maybe that would work. But otherwise, we've got Wolbachia is something that's used as an endosymbiont that makes males unable to reproduce with uninfected females. And then there's a number of different genetic circuits out there that either can make all of the offspring die, or say only the female offspring die, or only the male offspring die. And all of those could theoretically be adapted to crown-of-thorns, but they're based off of quite a few, basically a couple of decades of research into characterizing a lot of these genetic components, and it would take a long time to get a lot of these genetic circuits to work well in Cots. So more immediately, there are probably some other options. And those options are based off of some of these new possibilities that emerging from CRISPR. And for those of you who aren't aware, CRISPR is, initially it started out as a tool for just genetic engineering that's been expanded to a lot of other, it's a tool for genetic engineering, but the tool has been adapted in a handful of different ways so that it's a lot more versatile than it initially was. So on the left side, what you have is Cas9. So Cas9 is the main player in the CRISPR system, and it's an enzyme that's an endonuclease. It introduces double-stranded DNA breaks, but you can specify where those double-stranded DNA breaks are using a guide RNA. And a guide RNA is just a short RNA sequence, and there's a 20 nucleotide sequence that's part of that larger guide RNA sequence, and that through standard base pairing rules will direct the Cas9 wherever you want it to go in the genome with a few design rules, and it'll introduce a double-stranded DNA break. So that's a really great tool for introducing mutations, and then also for potentially knocking out genes, and there's a few other ways to apply it. But this has also been modified, and as I mentioned, in a handful of different ways. And on the right side, I'm showing a programmable transcriptional activator. And the way that these work is you have a dCas9, which stands for dead Cas9, meaning that its enzymatic activity has been mutated, so it no longer introduces double-stranded DNA breaks. Now it's essentially just a protein blob that you can send anywhere into the genome using a guide RNA. And that then has been fused to these additional transcriptional activation domains, meaning you can now use this to turn on more or less any gene that you want in whatever tissue that you want it in. So you have control over the transcriptome now as well. And using these tools a few years ago, I came up with a way essentially to engineer speciation events. And the way that that works is you would first use Cas9 to edit the promoter of some kind of tightly regulated gene. And the promoter is usually just an area upstream and adjacent to a coding region that regulates how and when that gene is expressed. And there's surprisingly a lot of flexibility in the amount of mutations. Small mutations you can introduce there that don't seem to have much of an effect. And in an organism that has that mutation, you then engineer it to express a transcriptional activator. And that transcriptional activator is looking for the original promoter sequence. So in what we're calling a synthetic species, you've got this thing that's just floating around more or less aimlessly. But if you have a mating with a wild type, then in the offspring, the transcriptional activator finds the wild type promoter and drives lethal overexpression of that gene, right? So if you have males that have these two mutations, you can engineer them you can release them and they'd be genetically incompatible with wild type. Is this gonna go? It's not. Oh, there we go. So we first demonstrated this in yeast. So I'll just mention real quick. On the left, you've got a permissive mating. On the right, you've got a non-permissive mating where we targeted actin, which is a cytoskeletal gene. And what you have in the non-permissive mating is so much actin is being produced that the cells literally just burst open. So this is definitely something that was effective in yeast. And then after that, we moved on to insects. So yeah, I'll just skip that slide. Let's go on to insects. And again, in insects, what we did is we targeted a few of these developmental morphogen genes where we were able to introduce mutations, express these activators. Those activators were then able to only affect those activators were harmless except when those insects, the synthetic species, mated with something with the wild type, in which case it was 100% lethality. And ultimately, we engineered 3 different synthetic species, which are all incompatible with each other and the wild type. So if we were to try to do something like this in Cots, the first step would be to identify some of these promoter targets, and one way to do that is to express this activator try to express this activator targeting various promoters and see what survives. And then once you know what survives, then there's a few different options, potentially depending on the technical challenges, where you mutate the promoter. Ideally, you'd be able to select for larvae that have the mutation and then use cloning to expand those larvae and then engineer them to express this activator. That results in chimeras, which you then have to cross to then get this synthetic species where you would just release the males. If you can't engineer if you don't end up finding a way to engineer the larva, then there's a longer process down on the bottom, where it can take a couple of generations. But that's where the high fecundity of the crown of thorns is pretty helpful. As far as the feasibility goes, we now, there's both of the papers here that I'm talking about. There's people in the audience that are responsible for this. We now have the genome of the crown of thorns. In sea urchins, at least, we have methods for genetic manipulation, and those methods, it gives us a starting point to now start looking at how to genetically modify crown of thorns. But to my knowledge, there hasn't been any genetic modification of the crown of thorns starfish. And excitingly, there's also this, some of this pheromone stuff that people have been talking about, and ways to potentially exploit that technology as well is to actually engineer the crown of thorns starfish to be expressing peptides that it's secreting into the water that are either causing manipulating the behavior of the other crown of thorns starfish, right? And so, for example, you can imagine having a crown of thorns starfish that is expressing alarm pheromones, or even it could be a gene from the Triton snail, and then it goes to where everyone's there having the big mating aggregates, and it just basically crashes the party and then disrupts those mating aggregates, and again could potentially be used to drive down the reproductive success of a particular population. And there's a few different ways to do this, again including cage or non-cage populations. And anyway, with that, I'll just thank some of the people that were involved in this in this research. Thanks.
Right. Thanks very much. I think that's a lot of information that can fuel our discussion. So.
Thank you very much, Sandra. And yes, please have a look at the Slido. There have been some really good questions put forward in that last session. If you were a presenter, you're more than welcome to email me the answers to the questions that you've been submitted, and then I can manually add them to the Slido for everyone to view. We're going to break for afternoon tea now. I think just because we are still running really behind schedule, we need to have everyone grab a coffee and then come back. And then if we do have time before the next session, we'll do a couple of questions then, I think. But I think now's a good time for us to just grab a refreshment and bring it back. So, thank you.